Welcome back, ladies and gentlemen. So the last topic that we are going to talk about this unit comes back to cancer. So when we talked about all of the coordination of your cells going through cell division, and now again talking about this coordination of cells becoming what they're supposed to be in terms of differentiation and gene expression, all of this comes back down to your genetics. And that means that we need to be able to use that set of instructions appropriately at the right times in order for cells to divide, in order for them to do their function, to structure themselves appropriately, to develop correctly. So, at any point along the way, anytime that we are controlling a particular system, that does potentially set up for the opportunity for mistakes to be made. And so anytime we have those mistakes, we have potential for cells to sort of go off the rails. And in this case, we're going to focus on more of the genetics of cancer as opposed to talking about things like growth factors and the density dependent inhibition and all of that sort of stuff that we talked about back when we talked about cell division, and just the fact that cancer loses that ability to control that cell division. So cancer really is the causative agent. It's the result of a bunch of mutations that happen to genes that regulate that cell division and that cell growth. That gene regulation system that goes wrong in cancer oftentimes are very similar systems, if not the same systems, that are involved in that embryonic development in terms of setting up body plans and things like that. So, how do those mutations get there? Well, some of them can be completely random, some of them can certainly be inherited, and we also know that viral infections, at least some of them, have connections to cancer in animals, including humans. For any of those of you out there who have had the Gardasil shots, you have been given a vaccination against human papillomavirus. And that HPV virus we're giving you not only to sort of help mitigate that virus itself, but most people who are infected with HPV don't generally have any real outward symptoms or signs of any particular disease. But what we did find is in um, women, cervical cancer was coming as a result of that infection. You may have been infected in your teens or your 20s, and then you're developing cancer from that in your 40s, 50s, 60s. And men, same deal, but with prostate cancer. So it's interesting to look at these viral connections. They're hard to find because you're often not seeing cancer pop up immediately after that viral infection. But that virus, in the methods and means by which it gets into your cells, takes over the host machinery to make more of itself, um, does cause disruptions to the cell's genetic mechanisms. So when we find these connections, it's often 10, 20, 30 years down the line, which is what does make it challenging to find the correlation between a viral infection and cancer, but we now think that there's somewhere on the order of about 25% of the viruses that infect us that may have cancer connections. And I'm going to guess it's probably even going to be higher than that as we continue to expand upon this research. So, when we look at cancers in general, there are generally two major classes of genes that we see become mutated that ultimately result in tumor formation. One of those genes are these things called oncogenes or proto-oncogenes, and they're basically the same gene, it's just one is a normal functional copy that we want to be there, and the other is the mutated version of it. So an oncogene is the cancer-causing gene. That is the mutated version of the gene. Where the proto-oncogene is the good copy, the one that we want to be there, and is responsible for controlling a lot of the cellular mechanisms responsible for that normal cell growth and division. So it really is the conversion or the mutation of a proto-oncogene into an oncogene that can lead to that abnormal stimulation of the cell cycle, what we would consider cancer. And a lot of these proto-oncogenes are there to produce those growth factors, right? So what we can see is when a proto-oncogene gets mutated, it can get mutated in a handful of ways, all of which sort of overstimulate that growth factor protein, whether it's in the production or we don't break it down fast enough like we're supposed to, or it just receptors become overly sensitive to it, so on and so forth. So one thing that can happen is some sort of translocation or transposition mutation, where they end up with a new promoter or a mistake in that promoter that ends up causing that gene to sort of be turned on all the time when it's not supposed to be. And that can produce over excessive amounts of that normal growth stimulating protein. We can get gene amplification. So if we go through something like crossing over, and it crosses over incorrectly, where now I have multiple copies of that same proto-oncogene on that chromosome where I should have had one copy. I've got 2 or 3 copies as a result of that. They'll all get turned on at the same time, and as a result produce more growth factor than they should. We can get point mutations in things like the control elements that regulate whether or not that switch gets turned on, and again, produce more than we should. Or it can happen within the gene itself, and now we can get a situation where that normal growth protein is either hyperactive, meaning it just sort of is overly sensitive, or overstimulates receptors or pathways in a cell, or it's degradation resistant. So there are going to be enzymes and other molecules that are going to come in and help break down that protein when it's no longer necessary because the cell shouldn't be dividing. And these guys sort of no longer fit quite appropriately into those enzymes and so stick around much longer than they should. So again, it can be a handful of reasons as to why these happen. This is kind of what I just talked you through. So movement of DNA within the genome, if it ends up near that active promoter, we can sort of upregulate transcription and as a result produce more growth protein than we should. Amplification, so increase the number of copies of the gene, and then those point mutations either within the gene itself or within the control elements of that gene, all which are going to increase gene expression and cause more growth factor. More growth factor we have just starts stimulating growth, exactly what it sounds like. And it should only be produced at appropriate times throughout the cell cycle. And this isn't what's happening here. It's being produced all the time or at inappropriate times. And that is where that uncontrolled cell division is coming from. The other subset of genes that we often see mutated are these things called tumor suppressor genes. So while the proto-oncogenes are supposed to stimulate cell division, tumor suppressor genes should be doing the opposite. They should help to prevent that uncontrolled growth. And they do this by sort of checking in on things. So they are producing proteins that may be in part of the damaged DNA repair systems, proofreading systems, ensuring that cells aren't going through that G2 checkpoint with damaged DNA. They can control cellular adhesion. So we talked about that anchorage dependence back in your cell cycle. Cells are not supposed to be able to divide if they are not attached to some sort of extracellular matrix, whether that be in a tissue culture or in your body. And if these tumor suppressor genes get mutated, that may be something that they lose. They are also supposed to inhibit the cell cycle in cell signaling pathways, so sort of listening to external signals from cells around them. So going back again to something like that density-dependent inhibition, these tumor suppressor genes, some of the proteins that they produce, are meant to be able to recognize, hey, I'm butting up against another cell. I've sort of filled that wound, I've filled that gap, and so I don't need to continue producing more cells. Mutations that decrease the tumor suppressor gene's ability to produce these proteins end up contributing as well to that cancer onset. So let me give you a few examples. So one of the most common examples that AP likes to use in terms of understanding proto-oncogenes is the RAS gene. And P53 is a common one they tend to use for the tumor suppressor. So mutations in that RAS gene actually lead to the production of an overactive or hyperactive RAS protein. So in this case, this is an idea of a signal transduction pathway. So we've got that growth factor coming in from neighboring cells. That's going to stimulate these receptors, which will then sort of run a cascading effect, sort of like dominoes in a chain, that then increases the transcription of a protein that stimulates that cell cycle. Well, in this case, we have an overactive or hyperactive RAS gene as a result of a mutation in its production. And now, rather than having to wait for that cell to get a growth factor from outside, it just gets turned on all the time. So in a normal cell that's functioning properly, this RAS gene basically remains inactive until that growth factor comes in from a neighboring cell and activates it. And that then leads this pathway to start. But in a cell that has a mutated RAS gene, that RAS protein gets locked in the on position. And so it's constantly stimulating this domino effect to constantly produce those proteins that stimulate the cell cycle, and it shouldn't be doing that. Suppression of that cell cycle can be really important in a cell with damaged DNA, and this is where that P53 gene comes in. So the tumor suppressor gene, the P53 gene, normally in a good functional copy, prevents the cell from passing on the mutations due to that DNA damage. However, if that mutation occurs in the P53 gene, we now prevent the suppression of the cell cycle. So that's what we're seeing here. So in normal situations, that DNA gets damaged, maybe something from UV light, and a good functional copy of that P53 protein that gets produced from that gene will then come in and prevent this particular damaged cell from going through the cell cycle. But if we end up getting a mutation in that P53 gene, it can no longer bind appropriately to the damaged DNA, and as a result, damage happens. Cell's just going to keep dividing. It doesn't know that the damage is there. And so we're going to perpetuate that problem of that DNA damage into the next line of cells and the next line of cells and the next line of cells. So think of it as something like a skin cancer cell. If a good functional copy of that P53 gene happens, you get a little damage in your cell from that too much sun that day. That cell sort of just gets stopped from going through cell division, probably goes through apoptosis, we destroy it, no one's the wiser. But if that damage not only does damage to the DNA somewhere, but also damages that P53 gene, I can't suppress that cell from continuing to divide, and now that skin cancer mutation continues to perpetuate into a actual tumor, and then we start detecting it as skin cancer. Here's the good news, I guess. When we look at cancer, it's not going to be you get hit by a single mutation, you develop cancer. It's going to take multiple mutations in order for you to get what we would really think about as full-fledged cancer, right, in terms of clinical diagnosis. The more rounds of cell division your body goes through, the more you have contact with known mutagens or carcinogens, the more likely you produce enough mutations to actually develop that clinical case of cancer. We're probably all sitting here with a cancer cell or two hanging out in our body because we've been exposed to something that has caused a mutation in a proto-oncogene or a tumor suppressor gene. But because we're not sitting here riddled with multiple mutations, most of us will probably never develop a clinically diagnosed form of cancer, at least not anytime soon. That being said, the longer you live, the more rounds of cell division your body goes through, the more chances you have to encounter those mutagens or those carcinogens. And so when we look at most cases of cancer, when do they typically occur in life? they're often in more older adults. So we need at least one active oncogene and mutations to several tumor suppressor genes before we often will end up starting to see these sort of typical signs and symptoms that are associated with cancer, tumor development to the point where it is actually recognizable on a scan, and we talk about clinically diagnosed cancer.
Final two things I want to talk about today is cancer and genes that protect against cancer. So essentially, cancer is a form of cell determination or gene differentiation. Different genes are being expressed, causing cells, cancer cells, or regular cells to become cancer cells. Two terms to know in this. An oncogene is a gene that causes a cell to become cancerous. A proto-oncogene is a healthy gene. So it's a pre-cancerous gene. What you see here on the right-hand side is they are showing you some sort of normal cell right here. So the DNA that is in the cell, it is presently a proto-oncogene. It is a healthy set of DNA. You get some sort of mutagen. Right here they're showing you radioactivity, and that breaks the DNA. Once that DNA gets broken, it's going to self-adhere or self-repair. It's going to stick itself back together. But when it sticks itself back together, it might do that in such a way that the genetic material gets severely broken, which would lead to this normal gene becoming an oncogene. That oncogene then is going to start doing all of the things that we talked about happens in cancer cells. You are going to lose anchorage dependence. You're going to lose density dependent inhibition. This thing could start secreting growth factors. The genes have become broken, and genes start being expressed that cause our happy normal cell to become a cancer cell that just replicates out of control and starts influencing all of its friends that are around it. Now, our body does have a guardian that you should know about. There is a gene called the p53 gene. It is also known as the guardian angel gene. What it is it's a tumor suppressor gene. It's present in the cells of our body, and it's essentially there to guard against broken cells getting out of control. It's like a regulator or a quality control gene or whatever. It essentially, like, if DNA breaks in the cell, it ensures that things get straight before that cell further divides. So there are a couple things that it can do. On one end of things, it can inhibit the cell cycle. So we talked about checkpoints in the cell cycle. If a cell gets to one of those checkpoints and the p53 gene says, nope, you are broken, it can cause a couple things to happen to shut down that cell cycle. It could bind to cyclin, and we talked about cyclin causing the cell to go into mitosis. So if p53 binds to cyclin, that cyclin's not there, our cell is not going to go along. So that gives the cell time to repair the broken DNA. It can also synthesize miRNA, which is microRNA, which can bind to DNA and slow down the cell cycle as well. So those two strategies are strategies p53 uses to give the cell time to let the machinery within the replication transcription mechanisms fix the DNA. If the DNA cannot be fixed, p53 sends the cell into apoptosis, which is programmed cell death. It says, nope, we're not going to get fixed. It would be better if the cell just dies. So it will go ahead and kill off that cell, and thus protecting us from cancer. I hope that this little tutorial on cell differentiation / differential gene expression was helpful. Thank you for joining us on the Lab 207 webcast. My name is Mr. Kite. We'll see you again.
So that's the interesting thing. So when you talk about somebody who has a family history of cancer, that means that they are born with some level of mutation. Now let's back this up a step. In order for cancer to be inherited, that means that a gene mutation somewhere along the way had to happen in either an egg or a sperm cell. That mutation now gets passed on to that offspring, and it now is in every cell of that offspring. And so when that offspring produces its egg or sperm, the mutation gets perpetuated on again and on again and on again. So if I were to develop skin cancer tomorrow, the likelihood of me passing that on to a future child is probably pretty low, right? It's occurred in a skin cell. But if somewhere along the way something were to happen to cause a mutation in one of my egg cells, now that potentially gets passed on to a future child is now in that child's every cell because my egg is part of that baby's first cell, along with dad's genetic information. That goes through the rounds of division, and now that baby, every cell contains that mutation. If they live in or survive long enough to have their own children, it gets passed on again. And so there are certain inherited oncogenes or mutated alleles of tumor suppressor genes that we see associated with certain types of inherited cancer. The BRCA1 and BRCA2 probably being ones that most people recognize. These are mutations that occur in your tumor suppressor genes and are connected with at least half of the inherited versions of breast cancer. So let's just say that hypothetically, I'm just going to pull out a number here, that it takes 5 gene mutations to cause breast cancer. Well, if you are a person who has no family history of that, that means you have to accumulate all 5 of those mutations somewhere along the way in your life to develop breast cancer. So it happens. But if you are somebody who knows that you have a family history for one of those mutations, so you get genetic screened, or you just happen to know your family history and you have a BRCA1 mutation, well, now you have one of those 5. Now you only have to encounter 4 more mutations before you develop the breast cancer. If you get scanned and you have both BRCA mutations, now you only have to develop 3. So the more inherited variations you are born with, the higher the risk you are at to develop those additional mutations and then ultimately develop the cancer. So again, it's not a guarantee, right? You still have to develop additional mutations, but the likelihood of you getting there just by living and encountering things that we know are just kind of around in our environment and hard to avoid that are carcinogens or mutagens, your chances go up. So not a guarantee, but you are at a higher risk, which simply means we get to ensure that we do a little more screening and catch things early if and when they do develop.